India stands as one of the world's most diverse nations, both culturally and genetically. A recent genomic study reveals that Indian populations possess a higher proportion of Neanderthal and Denisovan gene segments than any other global population. This ancient ancestry is further enriched by more recent genetic contributions from Iranian farmers, Central Asian steppe pastoralists, and South Asian hunter-gatherers, creating a unique and complex genetic tapestry. The story starts off just after 50,000 years ago, when a small founder population was mixing its genes with an even smaller number of Neanderthals somewhere in southwestern Asia. That small founder group then ballooned in numbers. Starting at only 1 or 2,000, it grew to tens and then hundreds of thousands. Some percolated northward into the steppes, diverting east and west to the ends of Eurasia. Getting an even faster start were a growing flood of descendants who entered Southeast Asia, hopping along coastlines until they reached Australia and Papua. This pathway led first across the Indian subcontinent. This inescapable geographic fact makes South Asia a key region for understanding the early history of both Eurasia and Oceania. Today, around 1.5 billion people live in India, with around 5,000 different ethno-linguistic and religious groups. Yet it remains underrepresented in genomic surveys, even when compared to other non-European groups such as East Asians and Africans. A new analysis of Indian genomes, the largest and most complete to date, helps untangle these groups' complex evolutionary history, uncovering a 50,000-year history of genetic mixing and population bottlenecks that shaped the uniqueness of genetic variation in South Asia. The analysis, led by researchers at the University of California, Berkeley, the All India Institute of Medical Sciences in New Delhi, the University of Southern California, and the University of Michigan, was published in the journal Cell. The researchers analyzed the complete genomes of 2,762 individuals, representing most of the nation's major linguistic, ethnic, and geographic communities. The Berkeley team found that most of the genetic variation in India can be explained by a single migration of humans out of Africa about 50,000 years ago. These populations interbred with now extinct relatives, Neanderthals and Denisovans, and then spread throughout Europe and Asia, including India. As a result, Indians and Europeans both carry about equal numbers of Neanderthal genes, between 1% and 2.6% of the entire genome. But what surprised geneticists the most was that Indians were found to carry a greater diversity of Neanderthal DNA segments than any other population in the world. Earlier studies of Europeans show that collectively they share about 30% of the Neanderthal genome. The much smaller sample of Indian genomes, however, contained Neanderthal ancestry segments representing 50%, half of the total, Neanderthal genome. And Neanderthal DNA wasn't the only trace of ancient humans found in India. The study also revealed segments covering about 20% of the Denisovan genome, again, more than what has been detected in any previous population. Priya Morjani, the senior author of the paper, explains, "This is because of the complex history of South Asians. They've had multiple mixture events over the past 10,000 years, followed by strong bottlenecks in many groups. Together, that leads to a very complex mosaic of different ancestries, such that when you compare the Neanderthal segments in two individuals, they're often not shared. Denisovan ancestry makes up only a tiny fraction of the genomes of most people in Eurasia. Yet, the story it tells is remarkable. Most of the Denisovan DNA found in living humans doesn't trace back to the same group as the Altai Denisovans, the population represented by the famous pinky bone discovered in Siberia's Altai Mountains, from which DNA was first extracted in 2010. That finger bone, known as Denisova 3, still provides the only high-quality genome we have from any Denisovan individual. Instead, most Denisovan ancestry in people today comes from other highly divergent Denisovan populations that once lived far beyond the Altai. The combination of those two factors has made Denisovan ancestry hard to study. One way scientists have tackled this mystery is by examining thousands of modern human genomes. In 2018, geneticist Sharon Browning, along with collaborators from Joshua Akey's research group, took a closer look at the faint traces of Denisovan DNA hidden within the massive 1000 Genomes Project dataset. Focusing especially on East Asian populations, their method allowed them to pinpoint and measure segments of DNA that could be traced back to Denisovan ancestry. What they found was fascinating. On average, East Asian genomes contained about 0.1% Denisovan DNA, roughly 1 part in 1,000. But there was more to the story. When they compared these genetic fragments to the only high-quality Denisovan genome ever sequenced, that of Denisova 3, a striking pattern appeared. Some DNA segments in living people were nearly identical to Denisova 3. Others, however, were far more distant, showing many mutations not found in that ancient genome. This suggested something remarkable. The Denisovan ancestry in modern humans didn't come from just one group. It pointed to multiple Denisovan populations, some closely related to the Altai Denisovan and others that had diverged from them long before. In the new study, led by postdoctoral researcher Alyssa Kuret-Knauf, researchers took a closer look at Denisovan ancestry across India. They found that people in the region carry on average about 0.1% Denisovan DNA, with values ranging from 0.03% to 0.117%. Much like earlier research, their findings revealed two distinct sources of Denisovan ancestry, one closely related to Denisova 3 and another that was far more ancient and genetically distant. These two Denisovan components appear in different proportions from north to south across India. This gradient reflects the region's complex genetic history. Over the last 6,000 years, India became a crossroads of migration, as early agricultural and Bronze Age populations mixed with steppe peoples from the northwest and East Asian groups from the east and northeast. These waves of movement, driven by trade, cultural exchange, and occasional invasions, left a deep mark on the genetic landscape of South Asia. Yet, the Denisovan legacy stretches much further back than the Bronze Age. In northern India, the research team identified ancestry linked to the Altai Denisovans, the same lineage represented by Denisova 3. This component may reflect gene flow from East Asia, since it is nearly absent from populations to the south across all of India. However, there is clear evidence of a second Denisovan influence, one that diverged long before the Altai branch. This lineage shares less than half of the derived mutations seen in Denisova 3, suggesting it split off hundreds of thousands of years ago. But who were these mysterious people? One possibility is that they were part of the D2 group, identified by Guy Jacobs and colleagues in their 2019 study of Papuan genomes. That Denisovan signal, which entered human populations roughly 46,000 years ago, can still be found today across parts of island Southeast Asia. Jacobs' team did not examine whether South Asian or Andamanese genomes share the same D2 ancestry, a question that remains open for future research. Another intriguing possibility is that South Asia once hosted its own distinct Denisovan population, a lineage that diverged from the Altai Denisovan long ago. If that's true, this region may have been part of a vast contact zone where Neanderthals and Denisovans overlapped and interbred, much like the evidence seen in ancient DNA from the Altai Mountains, where Neanderthal fossils showed traces of Denisovan DNA. It was here that scientists discovered Denny, an extraordinary individual whose parents were a Neanderthal mother and a Denisovan father, showcasing direct evidence of interbreeding between these two archaic humans. Whatever their origin, the traces they left behind are remarkable. Although Denisovan ancestry in India makes up only a small fraction of the genome, the total representation of Denisovan DNA here is greater than anywhere else on the Asian mainland. The research team identified roughly 500 megabases of Denisovan genetic segments, including many that may have had functional effects, particularly in genes related to immunity. A striking signal of enrichment of Denisovan ancestry in Indians is seen on chromosome 6. This region is part of the human leukocyte antigen, or HLA, complex, and plays a critical role in defending cells against viruses and regulating immune signaling. At this locus, the frequency of Denisovan ancestry is 9.4% in Indians, which is nearly 100-fold higher than the genome-wide average of roughly 0.1%. The researchers also found that Neanderthal ancestry has impacted several immune genes, including a gene cluster on chromosome 3 that impacts response to COVID-19. This region, present in 20 to 35% of Indians, increases the risk of severe symptoms after COVID-19. Of all the Neanderthal DNA identified in the study, over 11% of the segments found in India had never been seen in any other population before, unique fragments of a long-lost past, preserved only here. Another way to see it is this. India has retained more Neanderthal ancestry than any other region on Earth. In fact, more than 90% of all known Neanderthal genetic segments found across modern human populations are also present in India, meaning that less of this ancient legacy has been lost here than anywhere else. That's true, even though some East Asian populations carry a slightly higher percentage of Neanderthal ancestry overall. In their case, however, the number of Neanderthal DNA segments is actually smaller, but each occurs at a higher frequency. This pattern likely reflects founder effects that took place during the early settlement of East Asia. To put it in perspective, while more than 11% of Neanderthal DNA segments found in modern humans are unique to India, only about 2.4% are unique to East Asia. The researching team also found that the Neanderthal ancestry found in today's people was all consistent with a single extended pulse of mixture from a single population source centered on 47,000 years ago. The Neanderthal DNA found among more than 2,700 Indian genomes fits neatly into this picture. South Asia may have been one of the very first regions where early modern humans carried and spread their newly acquired Neanderthal heritage. The remarkable diversity of Neanderthal DNA preserved here suggests that the actual site of this ancient encounter lay very close by, perhaps in what is now Iran or Pakistan. From there, human populations would have naturally expanded northward into Central Asia and eastward across India and into Southeast Asia, carrying fragments of that Neanderthal legacy with them. The researchers behind the study also explored a deeper question: whether today's populations in India descend entirely from a single major out-of-Africa migration, or whether traces of earlier coastal dispersals might still survive in their ancestry. These questions are closely tied to long-standing debates in archaeology. For decades, some archaeologists have proposed a southern route for the spread of early modern humans from Africa. According to this idea, groups of coastal adapted humans moved northward along the eastern African shoreline, crossed the Red Sea, and followed the southern coasts of Arabia and India, eventually reaching island southeast, Asia, and Australia. This route was thought to explain how humans reached Australia before 50,000 years ago, long before they appeared in Europe. A related discussion centers on the Toba volcanic eruption, which occurred around 74,000 years ago. In the 1990s, some scientists suggested that this catastrophic event dramatically altered Earth's climate, potentially wiping out populations of archaic humans and paving the way for modern humans to expand from Africa. Because of its location, India plays a central role in both the southern route and Toba hypothesis. And for years, archaeologists working there have framed their research around whether the southern route existed before or after the eruption. But both ideas have evolved. Today, there is little evidence that the Toba eruption caused widespread extinction, and archaeological sites in South Asia show no major break in human activity at that time. Meanwhile, the southern route hypothesis has become intertwined with another question: whether an earlier, now extinct, out-of-Africa population may have existed in East and Southeast Asia before the major migration that occurred after 50,000 years ago. To test these ideas genetically, Karat Knarf and colleagues analyzed Indian genomes using minimum coalescence methods, essentially tracing when DNA segments in different populations last shared a common ancestor. Their results suggest that the ancestral population of India split from sub-Saharan Africans around 54,000 years ago. That timing matches other non-African populations, pointing to a shared founder event for all people living outside Africa today. Still, that estimate may be too recent. The coalescence approach simplifies complex histories, relying on assumptions about mutation rates and ignoring later gene flow that may have continued after humans first entered Southwest Asia. Small shifts in these factors could move the date slightly earlier or later. Even so, when compared with results from studies of Papuan ancestry, a model involving a roughly 10,000-year bottleneck followed by expansion after 44,000 years ago fits the data well. Crucially, there's no evidence that Indian populations contain a deeper layer of ancestry than other Eurasian groups. That said, the data cannot completely rule out a minor genetic contribution from earlier dispersals. The team estimated that up to 3% of Indian ancestry could in theory come from a pre-Toba population, but this figure likely reflects statistical uncertainty rather than solid evidence. The data are simply too limited to distinguish a small signal from none at all. These new findings represent a major step forward in understanding the dispersal of modern humans. The analyses strengthen the evidence for a shared founder effect, a genetic bottleneck that has already been documented across much of Eurasia, Oceania, and the Americas. Beyond genetics, the results also help address several long-standing archaeological debates. Excavations in both northern and southern India have revealed a striking continuity in stone tool technology, with techniques typical of the Middle Paleolithic appearing both before and after the Toba volcanic eruption around 74,000 years ago. Archaeologist Michael Petraglia and his collaborators have argued in studies from 2011 and 2020 that this continuity may indicate that modern humans were already present in India before the eruption. An alternative view, championed by the late Paul Mellers, proposed that modern humans only arrived in South Asia after 50,000 years ago, bringing with them microlith-based stone tools, small, finely crafted blades that appeared during a rapid dispersal out of Africa. In Mellers' model, this migration wave extended across Southeast Asia and into Australia. Petraglia's group, however, offered another interpretation. The emergence of microliths may have reflected population growth and environmental pressures rather than a new human arrival. Most archaeologists today favor a hybrid model. For instance, Shaila Mishra and colleagues suggested in 2013 a two-phase scenario. An early wave of modern humans reached Southeast Asia and southern China before 100,000 years ago, followed by a later migration, possibly from Africa, that introduced microlithic technology into South Asia after 50,000 years ago. 15 years ago, the genetic data available from South Asia were limited. Studies of mitochondrial DNA suggested that population growth began earlier and faster here than in other parts of Eurasia. In 2008, Quentin Atkinson and collaborators proposed a major South Asian chapter in human dispersal beginning before 50,000 years ago. Mellers viewed this as strong genetic support for his hypothesis, though others pointed out that uncertainties in mutation rates and limited regional sampling left plenty of room for alternative interpretations. At the time, it still seemed plausible that people with African origins could have reached South Asia well before 50,000 years ago, perhaps even earlier than 100,000 years ago. But the new genomic data tells a different story. They point to the rapid dispersal of modern humans into South Asia after the mixture with Neanderthals near the time of their arrival in India. These dispersing people quickly picked up additional genes from a divergent Denisovan group. This sequence of events fits best with a rapid dispersal into India after 50,000 years ago, rather than a long-established earlier occupation. Not unless those early modern people were living side by side with Denisovans throughout their existence, something we have no evidence for as of yet. The earliest inhabitants of India were hunter-gatherers, the ancient ancestors of South Indians, whose closest living genetic relatives today are the isolated peoples of the Andaman Islands in the Bay of Bengal. Genetic studies show that people from southern India retain higher levels of this deep ancestral lineage than those from the north. A major genetic shift began thousands of years later with the arrival of agriculture. Archaeological evidence, particularly from the site of Mehrgarh in present-day Pakistan, indicates that farming in South Asia began around 8,000 to 9,000 years ago. These early agricultural practices likely spread from West Asia as they involved crops like wheat and barley, first domesticated in that region. To trace the origins of these Neolithic farmers, Knopf and colleagues analyzed DNA from 14 ancient populations spanning Central Asia and the Middle East. Their results pointed to a close genetic match with 4th millennium BC farmers and herders from Sarazm, an archaeological site in present-day Tajikistan. Archaeology supports this connection. Evidence shows that Sarazm maintained trade links with South Asia, including with Mehrgarh and early Indus Valley settlements. One Sarazm individual uncovered during excavation was even buried with shell bangles identical to those found at Neolithic sites in India and Pakistan. These bangles were made from Indian Ocean or Arabian Sea shells. Later, another wave of migration added a new genetic layer. Around 4,000 years ago, steppe pastoralist ancestry, linked to the Yamnaya culture of the Eurasian steppe, entered the subcontinent. This influx played a key role in forming the ancestral North Indian genetic component and helped spread Indo-European languages. Today, this steppe ancestry makes up anywhere from 0% to 45% of individual genomes across India. Together, these three groups, ancient hunter-gatherers, early farmers, steppe pastoralists, forged the foundation of India's extraordinary genetic diversity. After this period of mixing, the genetic story took another turn. Over time, Indian societies began practicing strong endogamy, marrying within one's community or caste. This has been proven to help preserve the unique blend of ancient ancestries in Indian DNA by limiting gene flow and increasing the likelihood that ancestral genetic variations are passed down through generations. But while this has helped preserve certain ancient gene fragments, it has also led to an increase in the frequency of recessive genetic variants and disease-causing genes. One example of such a recessive trait is a mutation in the RYR1 gene that causes muscle paralysis and other severe reactions to anesthetics like mivacurium. It is particularly prevalent in communities such as the Visya in Andhra Pradesh and Telangana, but present at very low frequency across the rest of India, and not present outside India. The team also identified numerous rare and population-specific pathogenic genetic variants, including variants linked to blood disorders, congenital hearing loss, cystic fibrosis, and phenylketonuria. But this work is ongoing. Moorjani and her colleagues are continuing to analyze the Indian genomes in the study, which is part of the LASSI study that has collected over 70,000 individuals, and aims to sequence a subset to study epigenetic differences, metabolomics, and proteomics to understand aging and age-associated diseases in India. To summarize, India's DNA holds one of the richest and most complex stories in human history. From ancient hunter-gatherers to Neolithic farmers and steppe pastoralists, every migration left its mark. But what makes India truly unique is how much of our deep past it still carries: the most diverse Neanderthal and Denisovan DNA in the world, genetic echoes of humans who walked this land 50,000 years ago. Far from being a genetic crossroads, India is a living archive, preserving chapters of human evolution that have vanished elsewhere.